Thank you for stopping by our poster. Here we present our published work that studies the mechanisms underlying divergent IL-4 responses of genetically distinct macrophages. Genetic variation is a major driver of phenotypic diversity, disease risk, and signal response of cells. Previous studies have shown genetic variation leading to large differences in gene expression and signal response. However, the molecular mechanisms for how genetic variation results in these differences are poorly understood. In this study, we leveraged 5 different inbred mouse strains to understand the impact of genetic variation on macrophages in response to an anti-inflammatory cytokine called interleukin-4 or IL-4. Figure 1 shows an overview of our experimental design and the main dataset we generated based on bone marrow-derived macrophages from these mouse strains. We used RNA-seq to measure the transcriptomic landscape of macrophages in response to IL-4, and used a combination of ATAC-seq, H3K27 acetylation chip-seq, and RNA-PO2 chip-seq to determine regulatory elements and regulatory activities. As a result, we found highly differential gene expression across the 5 mouse strains in response to IL-4. Among the 693 genes induced over twofold in at least one strain, only 26 or 3% were induced over this threshold in all 5 strains. More than half of the IL-4 responsive genes were induced over twofold in only a single strain. On the level of gene regulation, we also saw large differences between C57 enhancers and enhancers of other strains. Such difference was scaled with the degree of genetic variation. BALB, NOD, PWK, and spread have an increasing number of genetic variations compared to C57, and therefore they have an increasing number of differential enhancers. Next, we applied our recently developed bioinformatic tool, MAGGI, to strain differential IL-4 activating enhancers, and we identified a dominant set of transcription factors required for enhancer activation. Here we further divided these strain differential enhancers into 3 categories, depending on whether the basal enhancer activity in the non-induced strains is lower or higher or equal to the induced strains. And that gave us the low basal, high basal, and equal basal groups respectively, as shown on the left of Figure 4. As the results shown on the right of Figure 4, we found that mutations in motifs of lineage-determining transcription factors like PU1 and CPP were associated with low basal enhancers, while mutations in motifs of signal-dependent transcription factors like EGR, STAT6, and PPAR-gamma were associated with equal basal enhancers. Of particular interest, the high basal enhancers were negatively associated with PU1. These findings were all later validated with our ChIP-seq data of these transcription factors. Finally, we tried to pinpoint the genetic variation that leads to the divergent IL-4 response of macrophages. We trained a convolutional neural network adapted from DeepC using our mouse data. Our training data consisted of 300 base pair IL-4 active enhancers and a balanced number of random backgrounds. Our model eventually achieved 0.89 for the area under the ROC curve on the testing data. We then used DeepLift to compute the importance of each nucleotide. Figure 5 shows a significant enrichment of DeepLift prioritized variants at strain differential enhancers, and such enrichment gets even stronger at a more stringent DeepLift score cutoff. Collectively, these findings revealed general mechanisms of how genetic variation can influence signal response of enhancers. Our results also showed promise of using natural genetic variation from mouse strains to evaluate variant interpretation methods based on deep learning. If you're interested in more details of our methods or results, you can go to our paper published in Science Advances using the link in the references section. Thank you for your attention.